Welcome to our course. I'm Dr. Trish Lawler, a research scientist and educator here in Birmingham. On our course, you're also going to hear from Dr. Zanya Stamataki, other scientists, and our fellow clinicians and pathologists based here in Birmingham. We are all based at either the University of Birmingham or the Queen Elizabeth Hospital, which includes one of the largest transplant centres in Europe. Liver diseases worldwide are increasing dramatically as a consequence of viral infection, obesity-related disease, and alcohol injury, amongst other things. For many patients, their only option is to have a life-saving liver transplant. This is a complex but fascinating procedure, which involves a multidisciplinary team of professionals. We perform about 200 transplants a year here in Birmingham, so over the next 3 weeks we will teach you about the science and medicine that makes it possible to take a liver from one patient and transplant it successfully into another one or even two people.
My name is Dr. Zanya Stamataki, and I'm a scientist and educator here at the University of Birmingham. We will start our course by illustrating the major functions of the liver and describing its anatomy and cellular constituents so that you can understand what goes wrong in disease. We will also talk briefly about the tests that doctors do to identify when someone's liver is failing and remind you of the major causes of liver disease. In our first week, we will also begin to meet some of the clinicians who treat patients with liver disease and see how the decision is made to transplant someone. We will consider some of the ethical issues surrounding organ transplantation and also meet one of our patients to learn what it is like to have a transplant. In our second week, we will meet some of the surgeons who perform the operation and learn a little more about the procedure itself. We will also learn about the very important roles of the transplant coordinators and what happens after the operation. We will also meet one of the transplant pathologists here in Birmingham to learn about what happens if an organ is rejected. Finally, in our third week, we will move to consider the future of liver transplantation. Here we will consider how the diseases that require a patient to be transplanted are evolving and how this can lead to clever technical solutions to increase the pool of donor organs available. Throughout our course, we will use videos like this to teach you. We'll also supplement the video with text documents where you can read more details. We have also added web links in our documents and our resources section so that those of you who wish to extend your knowledge have some good pointers to reliable sources of information.
There will also be plenty of opportunities for you to interact with your fellow students, for informal discussions and also to exchange information you found as part of our independent case studies.
So let's get started with week 1 and learn a bit more about your amazing liver.
In this video, we're going to revisit some of the structures we mentioned in our earlier activities so that you can get a feel for the gross structure of the liver, how closely it is nestled between the other abdominal organs, and more importantly, the joints which need to be made when a donor liver is connected to the vascular supply of the recipient. The idea is to give you a feel for some of the technical challenges the procedure presents to the surgical teams. In this video, we were fortunate to be allowed to use a fantastic piece of equipment that is available for students learning anatomy at the medical school in Birmingham. This is called the Anatomage, and it's similar to a giant touchscreen tablet or essentially a 6-foot iPad that is preloaded with medical image that you can interact with and modify in real time. We have 2 in Birmingham which were purchased along with some ultrasound machines and other teaching equipment thanks to a generous bequest from Mr. Kenneth Foster. I've put a link to the Anatomage website below this video so that you can find out more about it, and I must admit it was really interesting to be allowed to use it for a couple of hours. You can see here the Anatomage is preloaded with anatomical images and extra details like MRI scans and X-ray images that you can interact with to zoom in and out of key structures or to remove tissue layers like the skin, fat and muscle to see what lies beneath. We'll also occasionally show you Dave, our anatomical model who featured in our previous course on liver disease. He's about a third real size and allows us to orient you as to where we are on the body when we use footage from the Anatomage. You can also add and remove his organs to see how everything fits together, which is quite useful. Okay, so to start with showing you the potential of the anatomage, let's have a look at Dave. You can see nicely how the liver nestles in under the lungs. It's the dark brown kind of organ here sitting just above the stomach. The liver is on the left of this image and the stomach is on the right as Dave is facing towards us. If you were looking at your own organs, the liver would be on your right hand side. Now, I want you to imagine that we're taking a slice horizontally just under the lungs between the ribs so that we can cut through the thick part of the liver. Let's have a look, see what this looks like on the anatomage. Okay, so here we have another male where we've taken a slice across his tummy and he's positioned head down, so we're looking down into his body from the feet end. My hand comes in here and you can see how we can move and manipulate the images. Okay, so we have the spinal cord at the bottom with ribs coming out to envelop the liver around it. The liver is to the left of the image and you can see two major vessels which are appearing as sort of dark circles or tubes. We have the inferior vena cava or IVC that the blood from the liver drains into. This is dark red, and then slightly to the right and paler we have the aorta. You can also see some of the hilum where the vessels enter the liver. This looks like a hole in the centre of the tissue surrounded by sort of pale connective tissue. And also you can see large vessel branches throughout the liver. Most of these will be branches of the portal vein. And next to these will sit bile ducts and artery branches, but they're a bit too small to see in this image. You can also see the spleen on the left. It's unusually large actually, which makes me think that this person used for this imaging may have had a medical condition. You can also see the gallbladder, which is pale pink and hollow looking, to the right of the image. Also make the liver disappear so that you can see how the stomach, top right, is next to the gallbladder. The major vessels are also slightly more clearly visible now, and if we put the liver back in, you can imagine how snugly it fits in with all these other organs. And even more when we add back in the fatty tissue, muscle and skin layers. You could see how much depth of tissue a surgeon would need to penetrate in order to gain access to a liver for transplantation surgery. So we're going to illustrate this a bit more clearly in our next anatomage sequence. But firstly, back to Dave the dummy to illustrate the positioning of the liver in the abdomen again. Now, we're going to imagine our patient lying on his back with his head again toward the left-hand side of the image. So now we've removed the skin and subcutaneous fat to expose those beautiful abdominal muscles. Underneath these is the peritoneum, which protects all those organs in the abdomen and hold them in place. If you remove this, you can see the liver again, large and red on the left, with the darker gallbladder poking out from underneath, and also a bit of stomach visible. If we remove the liver again, you can see the stomach and esophagus where your food passes down into the stomach, going off to the top right of the image under the ribs. If we then move the entire rest of the gastrointestinal tract, you can see the kidneys situated towards the back of the patient. And they sit quite low down, just above your hips, and you can see the ureters which drain urine from the kidneys into the bladder. So if we use Dave to illustrate the position of everything again, minus the liver, you can see how faithfully our simple anatomical dummy recreates the images of the full body scan. We'll build back in the peritoneum and muscle layers now to remind you of how it's all held in place. So the next thing I would like to show you is from a different angle again. I've taken Dave's liver out to show you the back and the hilum where all the vessels connect into the liver. You can see where the hepatic vein would connect to the large inferior vena cava in blue on top of the liver. You can also see the gallbladder and common bile duct illustrated in green to the bottom of the image. So here's the same as a section through the anatomage specimen. And this time I've highlighted the vessels both within the tissue and as a box where that hilum would be. And you can also see the inferior vena cava as a dark red structure within this box. Now I'm going to show you this in a different way. Here we have our whole body image again. The head is top left, but now we're going to slice down into the tissue, towards the back of your screen if you like, so that you can follow the vessels through the liver tissue. I'll turn the image slightly and point to the area where the vessels appear. You can also see the bile duct nicely here, and also as the liver disappears, both the aorta, which is more to the right, and the inferior vena cava, which is more to the left behind the liver. You'll see this even more clearly in the next image, where instead of chopping horizontally across Dave's tummy, we're going to take a vertical slice from head to toe, and we'll move through the liver, up and down, to allow you to see the vessels. Arteries will be in blue and veins in red, while the lymphatic supply is green. And again, I will point out the hilum so that you can see how complex the vascular connections are between the abdominal organs. There's the hilum there. You can also see the large vessels there. You can see all the connective tissue around the hilum, and you can see the common bile duct entering underneath the liver there. The lymphatics are illustrated in green really nicely in this image. So to end this film, I've tried to show you how organised your abdominal organs are and how snugly they fit inside the body. So if we just turn our anatomical specimen here, we're going to end with this beautiful image of the vessels connecting into the back of the liver, so that you'll try and remember this when we chat to our liver surgeon about how organs are inserted and removed during transplantation. So if we just false colour this image now, there we go, so you can see the colour and you can see just where we've cut through that liver tissue. You can see the beautiful point at which all those vessels are coming in.
Hello everyone. In this introductory video, we'd like to give you a quick overview of the functions of your liver. If you are interested to explore liver biology in more detail, you might consider signing up for the next run of our previous course on liver disease, which was also hosted here on FutureLearn. In the meantime, we'll give you the basics here to help you understand what goes wrong when a liver fails and a patient requires transplantation. Your liver is your largest internal solid organ, weighing in at about 1.5 kilograms in an adult, although it does grow to fit your size. It is situated slightly to the right of your body under your rib cage and held in place by the diaphragm and intra-abdominal pressure. It's connected to the rest of your body by two main systems, the blood supply and the drainage of bile, which we will show you in more detail in later activities. Now we're going to zoom into a picture of a real liver. So if you don't want to see one, close your eyes for a second. So this is a picture of a diseased liver which was collected as part of the transplant program in Birmingham. Oxygenated blood from the body enters through the back of this liver via the hepatic artery. This is around where the blue arrow is. But interestingly, because your liver has major roles in digestion, there is another supply which also enters the liver from the abdominal organs. This vessel takes food materials directly from your intestines to the liver and is called the hepatic portal vein. Blood from both of these inputs mixes in the main body of the liver and then leaves via the hepatic vein for return to the systemic circulation. There is also a connection which drains bile from the back of the liver into the gallbladder. This is indicated by the white arrow, and the bile is stored here until it is required. This gallbladder is a kind of muscular bag, about 10 centimetres or 4 inches long. Okay, if you've had your eyes closed now, you can open them. So we touched on digestion there, so let's consider some of the main roles the liver fulfills for you. If we take digestion first, the liver stores and processes materials that you ingest. So sugars in your diet can be used for energy throughout the body, or they can be stored as a polysaccharide called glycogen within the liver tissue. This can then be released as glucose for fuel whenever necessary. Vitamins, minerals and useful fats can also be stored within the liver, or used here as building blocks for making useful materials. So the liver is a bit like a factory that makes useful proteins for your body. One of these is albumin, which is a very useful transport molecule which helps carry useful materials around in your bloodstream. Also, the proteins which help your blood clot, the so-called coagulation factors, are made within your liver. Your liver factory also recycles materials from elsewhere to make useful products. One of these, bilirubin, is made from the breakdown of old red blood cells when they are no longer needed. This bilirubin is transported to the liver bound to albumin, and you can see the blood vessels at the back of this liver in red in this picture. The liver then chemically modifies this bilirubin so that it becomes water soluble and can be transported into bile, which is stored in the gallbladder as we mentioned, until it is needed to help emulsify fats in your intestines. Then it would be squeezed and secreted along the common bile duct, which is shown in green in this picture, and into the intestines. Now this is important because many useful vitamins are fat soluble, and also many useful fats are found in your diet, so these need to be extracted to be used by the liver. Bilirubin can also be excreted via urine and faeces once it is made water soluble by the actions of the liver. The bacteria in your intestines also contain enzymes which can modify bilirubin in a similar way to help with this excretion. Now we've mentioned these enzymes, or proteins that help with chemical reactions, which are found within your liver and help processes such as the conjugation of bilirubin that I just mentioned. But there are other enzymes here too, which help to synthesize and break down fats and sugars for either storage or use. And other enzymes in the liver facilitate another of its important roles, the detoxification of chemicals and substances that enter your bloodstream or are generated in situ by metabolism. Prescription drugs, food additives, cosmetics, alcohol, any chemical substance is processed within your liver to break down and utilise anything that's useful for the body and to permit the excretion of waste chemicals into urine, faeces or bile. All of these complex processes are carried out by a variety of specific and very different cell types found within the liver. We'll learn more about these in the next couple of activities, but in the meantime, when you consider all of these important roles in digestion, detoxification, the manufacture of useful chemicals and bile production, it's easy to see how devastating it would be for your body if your liver stopped working.
In this activity, we're going to introduce you to some of the key cell types that reside within your liver. This is so we can explain what they all do and, importantly, how things change in liver disease. This will help you to understand some of the tests for liver function that we describe later on, and also some of the structures and cell types that our surgeons and clinical colleagues talk about later in the course. In particular, our liver pathologist, Professor Stefan Hubscher, will show you how the liver appears under the microscope when a transplanted organ is being rejected. By this, we mean when the patient's immune system recognises the foreign transplanted tissue and begins to destroy it. So it's important that we understand the basic structure of a healthy liver first. Let's start by looking at a healthy liver under the microscope. This is an example of a normal liver that was collected for transplantation but not used. This sometimes is because a clinical team noticed an abnormality in the donor liver, maybe an unexpected lesion or too much fat in the tissue that would stop it working properly, or there's some other functional indication that they are worried about. In this case, the donor's family may give consent for the organ to be utilised for research, rather than not being used. Generous donations such as this are one of the reasons that our research group in Birmingham is able to do the groundbreaking clinical research and teaching that we do. Now this section has been cut very thin, about 5 micrometers or 1/10 of the thickness of a hair, and stained with chemical dyes that allow us to see the individual cells. One of the dyes in this stain, which is called a Van Giesen stain, makes collagen or connective tissue appear red. You can see this indicated by the white arrows on this image. In a normal liver like this one, the connective tissue or extracellular matrix is there to provide a structure for the cells to sit in, and also to provide support for tubular structures like blood vessels. So there is not much red staining in this sample apart from around those vessels and bile ducts. The rest of the tissue stains a brownish colour with this dye, and you can see the nuclei of the cells, those are the bits which control the genetic information, and the cell itself, stained a slightly darker brown. I've also indicated some of these with white arrows here. So let's think about some of the specific cells you can see in this image. This liver has been stained with another common stain called hematoxylin and eosin to show you the hepatocytes. These are a typical epithelial cell or barrier cell, and they make up about 80% of the volume of the liver. They're sort of 20 to 40 micrometers across, which is about half of the thickness of a hair. These are the cells that make bile, store vitamins and fats, make important proteins, secrete glucose, and are also involved in detoxification and metabolism. Our next cells are another epithelial population, this time the biliary epithelial cells, which make up about 1 to 3% of your liver tissue. These cells are labelled with a white arrow in the image shown here. They look a bit like a string of beads. They make up the biliary channels or bile ducts, which can range in size from tiny little structures of the portal areas to great big tubes which drain out into your gallbladder. And these cells exchange materials to and from the bile, things like water and bile salts for recycling. You can also see 3 other major structures in this picture, one on either side of the bile duct and the big vessel below it. These are all blood vessels, and this area where they all enter the liver is called a portal tract. You can see 2 branches of the hepatic artery, which brings oxygenated blood into the liver, and these are indicated by black arrows, and also the portal vein below these. This is the big vessel which brings blood from the intestines to the liver. All of these vessels are lined by another epithelial cell population called endothelial cells. There are also capillary-like channels called sinusoids, which are another endothelial-lined blood vessel, there to ensure that there is a maximal exchange of blood contents to and from the liver. This makes sure that cells like the hepatocytes are abundantly supplied with oxygen, and also that anything they might make, such as the albumin and clotting factors we mentioned before, are easily secreted into blood. In this next image, we can see the edge of another of these portal areas, where the blood comes into the liver. But this time I've indicated the little group of cells that look like black spots in the bottom of the picture. These are white blood cells which sit inside the liver to protect it from any bacteria or viruses. So they tend to sit around those areas where the blood comes into the tissue to deal with any problems. There are several different white blood cell types which can either engulf or eat pathogens like bacteria to kill them, or they can kill infected or tumour cells, or they can secrete compounds which direct other cell populations to deal with a problem. In disease, the number of white cells increases rapidly in the liver to fight infection with something like a hepatitis virus, for example. These cells are really important in the context of transplantation, so you will hear more about them later this week, and also when we talk about organ rejection later in the course. Now, the next cell I would like to introduce you to is very important in a process called fibrosis that you may have heard of. These so-called stellate cells sit inside those sinusoidal capillaries I mentioned earlier, some of which I've indicated here with white arrows. The image next to the arrows shows the stellate cells, which are labelled HSC for hepatic stellate cells, and they're sitting next to a red vessel which is lined by endothelial cells, or EC. The stellate cells are star-shaped cells, hence a name, which sit between hepatocytes and endothelium like the filling in a sandwich. In health, they store useful fats or retinoids like vitamin A, which are used to regulate liver growth and function. This is indicated by the white bubbles in the picture. However, in response to liver injury, these cells change dramatically. They lose this retinoid and begin to proliferate fast, so they increase in number. More importantly, they secrete a lot of extracellular matrix proteins, like the red collagen we talked about in our first picture of the normal liver. Now, if you look at this next image of a seriously diseased or cirrhotic liver, this graphically shows you this build-up of connective tissue in red. As you can see, when those stellate cells are activated in disease, you get this enormous increase in matrix production, which compromises the rest of the tissue. There aren't many hepatocytes left to fill in all those essential functions that we described earlier, and also that build-up of scar tissue or fibrosis. It makes the liver very hard and shrunken and restricts the flow of blood through it. You can also see that this liver is quite inflamed, as there are a lot more of those white blood cells in this sample, which I've indicated by the white arrow. Now, this change in texture and function of the liver in disease causes a lot of problems that are commonly faced by patients with end-stage liver failure who need a transplant. The build-up of pressure in the liver forces tissue fluid out into the abdomen. This is called ascites and is very uncomfortable and needs to be drained regularly. The increased blood pressure or portal hypertension within the cirrhotic liver also can cause upsets in the blood supply to other organs such as a kidney, so then you might see renal damage too. You can also develop bulging vessels or varices in the esophagus in this situation, and these vessels can sometimes burst to cause dramatic blood loss in some patients. This is a very serious complication of cirrhosis. In addition, the lack of functionally capable liver mass due to that build-up of scar tissue where the hepatocytes should be means that key detoxification processes are not carried out efficiently. There can be impaired removal of toxic compounds like ammonia from the blood, and this can then travel to the brain and cause swelling and neurological symptoms or even coma, and this is called hepatic encephalopathy. Okay, so that's the major cells that make up the liver covered, and we've also touched on some key processes that we'll pick up on later in our discussions of liver disease and symptoms. In our next activities, we'll talk a bit more about both the plumbing of the liver, or the blood and bile supply, as well as key populations of immune cells.
Transplantation surgery involves cutting into your body, removing your ailing organ and replacing it with somebody else's organ that works as intended. Every step of this process can alert your immune system to take notice and respond. To understand how your body reacts to transplantation, we will discuss some basic concepts in immunology. Our immune system is complex and sophisticated. Imagine an army of cells derived from a common stem cell progenitor designed to defend your body from foreign invaders. Like any army, the immune system has soldiers of different talents and specializations. In this video, we will discuss the key players of the immune system, and we will introduce some new concepts. This information will help you understand the difficulties our bodies face during transplantation. Remember, you don't need to know all these words to follow the rest of the course. For those interested in learning more about the immune system, links to further resources are provided at the end of this page. The molecules recognized by our immune system are called antigens. These can be present in germs, such as bacteria or viruses, or they can reach your body through the ingestion of food. They are not always harmful, but by definition, antigens are molecules that can induce an immune response, usually an antibody response. We will talk about antibodies in more detail later. It is important to note that without the recognition of antigens, the immune system remains inactive, in patrol mode. When antigen is recognized, immune cells become activated. Another important concept is communication between cells. Activated cells release protein signals known as cytokines, which are biochemical messages that can be recognized by cells with specific receptors. Recognition of cytokines allows immune cells to communicate with other soldiers in their army and stimulates them to make decisions about whether to respond, stay put, or commit suicide by programmed cell death. This way, the immune system regulates the onset and the resolution of inflammation. The first line of defense of the immune system is known as innate immunity. Although highly effective in eliminating many pathogens, innate defenses are not specific and they do not offer long-term protection from infection. Immediately following injury, neutrophils are among the first cell types to rush to the site and destroy bacteria. During this process, a lot of the inflammatory chemicals are released, and these recruit other immune cells to the battleground for assistance. Neutrophils and other cell types often kill pathogens by eating them, using a process known as phagocytosis. This word in Greek means eating a cell. Immune cells recognize pathogens directly or using the help of antibody molecules that can be captured by specific receptors. The captured pathogen then becomes engulfed by the immune cell, which ends up digesting it in acidic compartments. This could be the end of the line for a pathogen. Alternatively, we move on to more sophisticated cells, like dendritic cells and macrophages, which can use the ingested pathogens to fuel the activation of more deadly immune system warriors. This process is known as antigen presentation. Antigen-presenting cells are capable of breaking down pathogens into tiny fragments which can be associated with cellular receptors that bring them back to the cell surface. These receptors are known as major histocompatibility complexes, or MHC, and you can see them here in number 5 of this diagram. These MHC molecules are very important in the context of transplantation because they are highly polymorphic. This means that there are multiple variants of each gene within our population. The MHC genes are in fact the most polymorphic genes known. To be able to not recognize another person's organ as foreign, transplant donor and recipient MHC molecules need to be as closely matched as possible. Also, MHC molecules are polygenic. They are made from several different genes so that every person has a set of MHC molecules with different ranges of specificity. This way, many antigens can be recognized and presented to the most specialized cells of the immune response. The specialized arm of the immune response is also called adaptive immunity and is characterized by slower responses which are highly specific for particular antigens. B cells are multitaskers. They are professional antigen-presenting cells. They make cytokines that orchestrate other immune cell function, and they differentiate into antibody-producing factories. Antibodies are a critical part of the adaptive immunity. They are molecules that are released to seek and bind foreign antigens and mark them for destruction by phagocytosis. Cytotoxic T cells are the main effectors that wreak havoc and destruction. When they recognize antigen on MHC molecules, they release enzymes that kill the presenting cell to eliminate infection. They are effective serial killers and leave dead cell debris behind for phagocytes to clear away. Helper T cells also recognize antigen on MHC molecules. They then become activated, proliferate, and release appropriate cytokines to instruct the body's arsenal on whether an antibody-mediated attack or a cytotoxic attack would be more appropriate. Natural killer cells are both part of the innate and the adaptive immune response. As their sinister name suggests, they can also kill infected cells that present foreign protein fragments on their MHC molecules. NK cells have activatory and inhibitory receptors on their surface, which help them decide whether to kill or spare a cell. As you can imagine, all these trigger-happy defender cells must be kept under strict control to avoid damaging our own healthy cells, particularly in sensitive areas of the body. The liver sees a lot of non-harmful food antigens that arrive from the gut, so it is important that immune responses are dampened to avoid chronic damage. Regulatory B and T cells take on this task, and they do this by producing cytokines that can suppress T cell effector function. There are many regulatory cells present in the liver. When regulation fails, liver injury can lead to hepatitis. Regulatory cells are considered as potential therapies for liver inflammation that goes on unchecked, such as in autoimmune conditions. We hope you enjoyed this taster of immunology. If you would like to know more about the intricacies of our immune system, please follow the links below.